Great. So this is Diane Kaplan, and my project is finding Intron splice sites. A little bit of background of the problem. So for eukaryotes like ourselves, DNA sequences contain lots of introns, probably just as much as protein coding DNA. So these introns, it's not translated code. So when we make a protein, the gene is first transcribed into RNA, and then the introns are spliced out, the exons are stitched together, and the remainder is translated into the protein. Therefore, if we want to get to the functional importance of a sequence, we need to be able to strip out those introns just like the body does. So some challenges. In a long sequence, how can we successfully find the boundaries of introns? We have differing splicing motifs. Splicing motifs are not always the same. There is a lot of consensus, but there are different splicing motifs, even within the same creature, in different genetic regions, different tissues, and even different stages of development. Then, to make matters worse, we have alternate splicing. So back in the good old naive days, we thought that DNA translation was as simple as one gene making one protein, but then we saw that we don't have nearly enough individual genes to work it out that way, for eukaryotes especially. For 20,000 protein coding genes, our number of distinct proteins is magnitudes higher, 100,000, 300,000. And it turns out the genes can actually be made up of exons from different locations in the genome, and different combinations from different combinations of the exons can make different proteins. So yeah, they can use different reading frames, different strands, they can overlap with other genes. So interpretation of these sequences absolutely relies on correct splicing. So we're not leaving anything in that isn't actually there at the time of translation, or taking anything out, screwing up the reading frame. It's important to get this right. So it's been done already. These first three bullets are a few of the tools that have been used. This info tool enhanced identification of small exons, as small as just 30 base pairs, and better delineation at the junctions. Both of these second tools are available online and easy to use, especially this human splice finder tool, which we'll see in a couple minutes when I demo. And then here I called out three, but there have been a lot of little steps along the way, finding trends that'll help us identify what's where. So at Rockefeller University, researchers found that there are different nucleotide frequencies in different genetic elements, like promoters, exons, introns. So that can provide a good clue for where splice sites may be. The ENCODE project, this is a follow-on to the Human Genome Project, basically trying to identify all the functional elements in the human genome. Their work is ongoing, but in 2007, they made it known this part about one stretch of DNA making multiple overlapping RNA. And then some Danish researchers noticed a few years ago that genes that have a lot of introns in them have stronger splice site signals for the introns toward the ends of the genes than in the middle, sort of a reverse bell curve. So yeah, but looking online, I've seen a lot of different little clues like these that could be different directions people could take to help enhance our ability to predict where these sites are accurately. So my plan of attack, start with the basics and fine-tune from there. So This should be familiar from the textbook. Here are the logos for splice site motifs. So as a review, from the 5' end to the 3' end, the part where the intron starts is called the donor site. And then after the intron, the part where it kind of stops to be exon again is called the acceptor site. And these black lines delineate the exon from the intron. So when we find these motifs in our sequence with a good enough score that we believe it, we would keep the sequence, we'd truncate here, and then we'd pick it back up again here and keep going to have just the good stuff, just the exons. So to build my splice site finder, I started with these 4 functions. First, I want to be able to determine whether a bit of the sequence is likely to be a splice site. So I made 2 libraries, one for donor site and one for acceptor, with the probabilities for each base happening at each position for that motif. I passed to it the little snippet of the sequence to evaluate, and I also pass which type I'm evaluating it as, and it calculates the probability of getting that little bit, getting that NMR in the state of the motif that I'm asking about versus happening by chance given the usual frequency of each base in human DNA. I have a 3rd library for that. And if it's better than by chance, it returns the probability for further evaluation. So this is just to make an initial candidate list to consider. Second function is basically just to do that. Go through the sequence, you know, character by character, take little bits and pass it here to see, you know, whether it could be one of these. So I want to save a list of donor candidates, save a list of acceptor candidates, along with their scores and their positions in the sequence, so I can go back and do something with them later. 3rd function basically takes those lists and picks the winners, returns the location and the motif type with the highest score. And then finally, once we have the correct list, asterisk, asterisk, we can take a sequence, we can take our list of qualified donor and acceptor sites, their locations, and return back a cleaned up sequence with no introns ready for analysis. So my results so far. First we'll demo my program versus the human splicing finder. So let me minimize this. All right, so I'm going to use this CDK10. It's a gene that has 10 exons. I'm going to pick the second one just so we know we're already in the middle, so we'll have an acceptor site and also a donor site after. So we're starting with an intron, then we get to the exon, then we come back. So I'm going to take that guy and, oops, wrong one. And go here, follow the spectacular button. I'm going to do analyze the sequence, pick a gene name, this guy, and I'm going to choose exon 2. And first, just so we can all get our bearings, the way they notate it here, I don't know if this is common and everyone already knows this, but all the lowercase, the bases that are shown in lowercase are intron bases. Then when we have capitals, we've gotten to the actual exon 2, and then intron again after that, just so we're familiar. And it's starting AAGA, etc. So just for this code, I have that grabbed, and I'm going to, yeah, just get the list of good donors, acceptors, and the winners. So I'm gonna run it and give us some of them to see what we have. So these are the lists of positions, what it was, and the probability score I gave it. In the acceptor motifs, you can see this guy is the highest score, 8 compared to the other ones. And then in this list, this one doesn't even have an E value, so he is a clear winner. So we have a winning acceptor site at position 89, winning donor site at position 171, and we'll compare that to our results here. Now they've got a much fancier algorithm going on than mine. They've got more results, and they've got a whole different, this consensus value is a whole different ballgame. But the way they notate here, this is the winning acceptor motif at position 89, and it's the same one we had there. Winning donor, we want to be at 171. There it is. And all of my pretty good guesses that came back are in this list too. So yeah, that part was pretty thrilling actually. And then I'll do a quick demo of just the part where you provide the sequence, provide the splice site locations, and get back your clean sequence. So I chose a sequence that we all know and love, which is the alphabet, with 3 big fat introns of varying lengths in the middle there. I have my positions of where the donors and acceptors are, and I'm going to run it, make clean seq. And I run it, and it gives us the alphabet with none of the other junk in there. So yeah. Alright, back to presentation. Okay, so what's next? A lot. So I've been testing with a set of 7 exons so far from 2 genes. My initial primitive approach finds the winning sites for 4 of them, but in the other 3, I have some other motifs that score just as high but are not correct. So I could improve my scoring probably a lot of ways, but first that come to mind, I could add weighting for the positions, not just probabilities, and I could make use of some of the other trends that were noted in some of the stuff I've read, and one of them mentioned before about the different base frequency in different regions. Another thing would be making a threshold. Instead of just finding what's the best one in this list, because obviously that only works if you're looking for exactly one of each, and I don't want to have to know that. I want to be able to take a big sequence and find the right ones even when there are lots of them. So I want to work on that a little bit. I would also like to look into making it faster. I haven't really put it to the test with big long sequences yet, as I just get it up and running and working, but I'm excited to do that. And then also testing with a broader range of samples than just these human genes. And here are some of the references I mentioned, as well as where I got those 2 images. And that's it. We will take questions in class itself. So thank you. Oops.